There we are. So as I mentioned earlier, a couple of things that are sort of on our agenda to-do list. The first is, of course, that for you to complete your first quiz. Your lecture quiz is due tonight at midnight. The second lecture quiz is going to be scheduled for March 2nd, which is 2 weeks from today, and that will include chapters 8, 9, and 6. So 8 and 9, again, should be review from Bio 1 with a little bit of new material in 8 for some of you, but not for all of you. And then 6 is basically the new material, the new material about viruses, probably a little bit more detail than you've covered in Bio 1 before. So that quiz comes very quickly. I was hoping that today we would spend some time reviewing chapters 8 and 9, or at least most of chapter 8. I would remind you to, again, watch the videos, print the note outline, fill them in, read the chapters, particularly from chapter 8 with the enzymes, with metabolism, right? So the chapter's kind of broken down into the whole first part of chapter 8 is about just about enzymes and how they work. So you need to review that from Bio 1. Things like how they're regulated, what the active site is, their structure, their function, how they lower the action free energy necessary for a reaction to proceed, all of those things you need to remember, including how they're regulated, both in the laboratory and inside a real cell. And then the chapter launches into metabolism, which will break down into glycolysis, the prep step, the Krebs cycle, which also known as the TCA cycle, right? And then the electron transport chain. And I like to call it the electron transport chain plus ATP synthase. You may have heard this called before as chemiosmotic coupling. I don't usually use that word. It's not really that accurate. And again, all of those steps, what you need to know for all of those steps is outlined in the note outline, but I'll just review it here as well. You need to know the starting and ending molecule for each of the steps. And you don't need to be able to recognize it, but you should know its name. You should be able to tell me the starting molecule and ending molecule for each step. You also need to be able to tell me for each of the steps. You need to keep track of the following molecules, meaning how many are produced or how many are consumed in each of the steps. And for that, we're going to be looking at ATP, NADH, carbon dioxide, as well as FADH2. And I can't subscript on the whiteboard on the Zoom page, so you'll just have to not the water. We're not following any of the water. Okay, so for each of the steps, you need to be able to tell me the production, how many the production of each of these molecules. And then the last thing you need to be able to tell me is where in the cell. And we need to know both for prokaryotic cells and eukaryotic cells. So for eukaryotic cells, that should be review from Bio 1. Prokaryotic cells, it's basically everything happens in the cytoplasm until we get to the electron transport chain and ATP synthase, and then that is actually in the plasma membrane or the cytoplasmic membrane or the cell membrane, whatever you want to call it. And then there is an extensive discussion about fermentation and anaerobic respiration. So for that portion, I would pay particular attention to the video. Those are two separate things, fermentation and anaerobic respiration. I know in anatomy and physiology, a lot of times your professors do not distinguish between them, but they are two very different things in bacteria. Fermentation can occur in the presence or absence of oxygen. Anaerobic respiration is usually only by organisms that are anaerobic, and that is, of course, in the absence of oxygen altogether. Then the last little thing there is in this chapter, there's a little bit about the integration of metabolism. meaning how proteins are digested, how lipids are digested, how they feed into the various parts of glycolysis and the Krebs cycle in particular. And then there's a whole section about photosynthesis. And while I love photosynthesis, this is a skip for us, right? We're not going to be spending a lot of time talking about photosynthetic bacteria, so you can skip that section entirely, okay? That will not be covered on the next exam. That's pretty much all of chapter 8. So if you think back to Bio 1, that was essentially chapters 8 and 9 in your Bio 1 textbook. An introduction to metabolism, which is included here with the enzymes. And I would remind you about, let's see if I get my box. I can't add anything there. Okay. But I would also remind you about molecules that are reduced versus molecules that are oxidized, because that oxidation reduction reaction is what's powering the production of ATP. So I would remind you that from chemistry, as well as from the beginning of the chapter, they talk about the reduced and the oxidized form of NADH being the reduced form. So yes, you do have to know that as well. Chapter 9. That's quite a chapter, right? So that's all of DNA structure, replication, and then transcription. So how we make RNA, and then translation, how you make proteins from that RNA, how amino acids are put in the polypeptide chain. And then there's a section on gene transfer. There's also a section on mutations, and there's also a section on gene regulation. For our purposes, we're just going to focus on the lac operon for that. Again, some of you may have gotten that far in Bio 1, some of you may have not have. I personally usually don't talk a lot about gene transfer in my Bio 1 class or the lac operon at all, but the rest of the material, the DNA structure, the replication of DNA, all the steps, all the enzymes, the order of how it happens, how the leading strand is transcribed or replicated, how the lagging strand is replicated. Transcription, translation, again, the enzymes, the steps, translation, the 5 steps, how that ribosome assembles, where the messenger RNA is in that ribosome, how the peptide bonds are formed. And then gene transfer, a lot of times you'll see that called horizontal gene transfer, meaning between bacteria, since bacteria do not reproduce sexually, right? So they reproduce asexually. So a way for them to increase their genetic variation is by transferring genes to their neighbors. It's usually within the same species of organism. And this would include, I usually do them in the order of transformation, transduction, and transformation, conjugation, and then transduction. Transduction, for our purposes, since in chapter 9, you'll notice in the video that I talk specifically about, you need to know the sort of like the basics of transduction, particularly what we would call generalized transduction. Sorry, transduction. You'll learn more about specialized transduction in chapter 6 when we learn about viruses. And then the last that gene transfer are transposons. Again, all of that and all of this should be reviewed from Bio 1. So again, there's a few new things. Like I said, this fermentation and this portion here, the fermentation and anaerobic respiration, particularly the differences and similarities, as well as for some of you, the gene transfer and the gene regulation will be new, but the rest of it should all be refresh or add a little bit to what you already know. So those lectures are quite extensive. So they're here. You'll notice for chapter 9, there's actually 3 lectures for chapter 9 because that chapter is very thick. The metabolism lecture is a single lecture, but it is really long because I just went sort of powered through the whole chapter. So this is going to take you a while to get through, as well as the homeworks for chapter, I don't think there's any I guess this is just this is all the homework we have, right, for chapter 8. I might break that up into a few pieces because I think it's really long. So if that changes, those of you that I probably can't do that now because some of you probably already started it, but I'll take a look. And if I can change chapter 8 into a few smaller bites of homework, I will. If people have already started it, then I can't. So I'm just putting that out there. Chapter 9 is broken down into 3 parts. And then chapter 6, the video is fairly short. There is a note outline for chapter 6. And then I did break it down into 3 homeworks that are fairly short. Again, because this chapter 8 is kind of a long draw on on the time that you have to sit down and do that. And I know people will complain, but it is what it is. As I mentioned, so these labs are repeating. You don't have to do them twice. They're in here because that's where they were from last semester, as well as some of you may have already started this module before you took the quiz. 1 from chapters 1 through 5, that's why they're in here as duplicates. You can't do them again and get a different score, right? The virtual labs you just have to do until you get 100 basically. But I will be adding more virtual labs, and as mentioned, this exam 2 here, this lecture quiz 2 is going to be on March 2nd. And then we will also be adding some of the real microscope labs in this module as well. All the microscope labs, we're going to try to complete those prior to spring break. And then after spring break, we're going to, I'm going to ask you to rehydrate your bacteria and then begin your, and that's sort of self-paced. Once you rehydrate your bacteria, don't throw anything away, but you'll have to kind of self-pace, and it will be based on how fast they grow in your house, basically, so that when you can move on to the next one. So I think it was Hannah was asking, if you check down here to the very bottom, you'll see the Carolina. I have 2 modules on the Carolina labs. These are, the first one is lab safety in the microscope. This will probably change a little bit, the lab report. You will probably turn in 1 lab report with some drawings for the microscope, and you will have to do the lab safety, watch the lab safety videos and take a lab safety quiz. And then the module 2 for the lab kit is all of the live stuff with the bacteria. And then these videos that say video Carolina lab kit rehydrate cultures, that's me explaining to you how to do it. Day 2 is isolation, right? After I've got the bacteria growing, and you'll see me labeling the plates and doing everything step by step. So those are the videos you're going to want to watch and then follow along the procedure, right? The procedure is also in those lab handouts that are in the lab kit, this first lab kit module. So see where it just says lab kit experiment 2? So that's just the handout of looking what the experiment actually is, and it'll show you, oh, you have to download them and save them. Sorry, I can't show you from here. But it just tells you, it's like, looks very similar to a lab manual in that you would flip through and follow the procedures. You'll have to read those first and watch the video. These are some tips on how to do things. And then when you go back to the module itself, that's where the videos, I've tried to put the handouts, the words with the actual video of me doing it, right? So this is actually lab kit day 1, video 2, lab day 2, aseptic isolation, day 3. I think you can do these the same day. That's why, or you can split them out, doesn't matter to me. This is important that once we get to that portion, you read, because you will have to kind of plan out how you're going to do your labs, right? And of course, the schedule is going to be flexible. Again, it's going to be based on how quickly the bacteria grow in your particular environment. So I made out a sample schedule if everything was perfect and I had an incubator that was at 35 degrees Celsius, all the steps I would do day after day. In reality, it probably took me closer to a week and a half to 2 weeks to complete those. Again, which is why we're going to do those after spring break. And then there's also some virtual labs that we'll be adding in from Connect in that last section as well. So, but the microscopes, we're going to start microscope work probably next week, because I'm going to say that I'm posting an announcement that if you don't have your lab kit yet, you need to order it, because we're going to begin, and the first microscope lab is going to be due on the 26th. So I'll put together some the lab safety, make sure, move that up to the module so you can see it, duplicate it, put it in this module. The lab stuff will always be all together in those 2 or 3 lab modules at the end, but then as we do the activities, I'll probably duplicate the particulars and put them in each of the modules, because I get it, students have a hard time finding everything because they can't scroll through and concentrate. So that's that. The microscope lab will probably be 1 or 2 short lab reports to turn in. You will have to upload either photographs or documents. So I would advise being able to either print something and then scan it, or being able to photograph and add photographs to your document. So you that'll be a new skill for some of you that you'll have to work out, but those will start to be due. Like I said, we're going to finish the microscope work as best we can prior to spring break, and then we'll do the actual growing the bacteria. And then there's a little more microscope work to do with that after spring break. Other questions? Let me go back to my whiteboard, see if it's still there. I think sometimes, oh, it is still there. Okay. Other questions from chapters 8 or 9? Yes, no, maybe so. And then next Tuesday, we'll have a chance to talk a little bit about chapter 6. But again, next Tuesday, you will, next Tuesday we can talk about 8, 9, and 6 if you have questions about 6. And then again, the next lecture quiz is going to be scheduled for March 2nd. And it will include 8, 9, and 6. All right, so there was one more thing I wanted to check for here. So it looks like there's only a note outline for chapter 6. For chapters 8 and 9, there is not a specific outline. It's read the chapter, watch the videos, and take the notes. So I'm trying to force you to write the notes while watching the videos, because that helps you actually learn it and remember it. And then I think, yeah, for chapter 7, there's a note outline, and for 11, there's a note outline, and for 12, I think there's one as well. That's PowerPoints. Let's see. Yeah. Yeah, looks like there's 2. Those are the homeworks. So maybe not for 12, but here and there, there's still some note outlines. All right, questions, comments, worries, concerns. And it can be anything, even for the stuff that you're supposed to be already knowing for your quiz that you're supposed to be taking today. I noticed that only about 5 people have actually taken the quiz. So hopefully everyone's not going to try to do that at 11 o'clock tonight, because that will be problematic for getting onto Canvas. So I would try not to put that off if I were you. Go back to the whiteboard, because if I leave the whiteboard, then it's going to disappear. So that's all right. I guess I'll just leave it.
For the lab kit, it wasn't super clearly labeled. I ended up just putting everything into the fridge, minus like the microscope, of course, and like the dishes that had nothing. Is that fine if they're just all in the fridge?
It is. I did send an announcement with specifically what should go in the fridge that actually must go in the fridge. So you should have a rectangular box that opens from the top that has a plastic bag in it with 10 Petri dishes. That needs to go in the refrigerator. You should have 2 skinny boxes that are about yay tall and thin, and inside of them there are cardboard cutouts so you can put those. So the test tubes are in there. There should be 2 boxes of those, one that's broth and slants, and one that is slants and deeps actually, and one that has the triple sugar iron agar in it. Both of those boxes need to go in the refrigerator. And then there's a plastic bag that has the bacteria in it. So you need to sort through the plastic bags, and one of them has the bacterial cultures and it's small. It has a metal band on the vial and there's a little rubber stopper in there. Those need to go in the refrigerator, as well as the reagents for the biochemical tests, which would include the oxidase reagent and the hydrogen peroxide, and they are in very small dropper bottles. The stain set does not need to go in the refrigerator, and I wouldn't put it in the refrigerator because it's going to cause the crystal violet to precipitate, and then it won't work very well. So no, don't put everything in the refrigerator. There's a list that I put in the last announcement of what to put in the refrigerator, because people were telling me that they got, what should you refrigerate from your Carolina kit, right? All the media, the dehydrated cultures, and the biochemical testing /kit /bag, whichever, it's in a small plastic bag that contains the oxidase and the hydrogen peroxide. Do not put the stains in the refrigerator. That's no good. Get them out of there if you put them in there.
Okay. I just put everything, but I'll take it out because I got it before the announcement, and the only thing labeled to go in was the agar plates.
Right. If you don't put them in there, they'll all have mold on them by the time you go to use them at the end of March. So, I mean, I don't know if they changed the directions. Like I said, in the announcement, I'm not getting a lab kit this semester. Last semester, I got my own lab kit. This semester, I'm not getting one, so I can't see what you see. And students said that different kits, that when they talked to their friends, different kits said to refrigerate different items, which I really don't have any basis for that, but maybe, right? The media, all the media definitely, and the bacteria, the oxidase and the hydrogen peroxide have to go in the fridge. The rest, room temperature is fine. I would assemble your little test tube rack and I would open the bags and kind of see all your materials, what you have. Obviously, right, the microscope comes separately in its own little box. Those plastic slides we'll use to make a few slides, but there are glass slides included in the in some of those bags inside the rest of the kit. There's actual glass slides, and when we make bacterial slides, we won't use the cover slips. So it's kind of a 2-part thing: the microscope and a few slides to look at and a few slides to make, and then the actual sort of microbiology portion.
Yeah, when I opened my kit, I saw the one pair of test tubes I put in the fridge, and then I put the discs in there, and then I just like went through all the bacteria and just read it and just said keep cool. So I was like, okay, obviously that goes in the fridge.
Yes.
So
Yes.
that's pretty much what I did, and then you posted that, so then everything you said mostly was in there except for I think like one thing, but yeah.
Yeah, the oxidase of those reagents, the oxidase is probably the most important to put in the fridge. The hydrogen peroxide will go bad if you don't. I mean, it'll work, but not as well. So like I said, since we're going to be holding those kits for about a month before you actually start doing the bacterial work, it is going to be important that the media is refrigerated. Otherwise, it will go bad, and then you can't actually do the experiments. So yeah, get that stuff in the fridge. Also, as recommended, I would be sure that you check that you have everything. They're pretty good about sending out replacements if something's broken. All right, questions about the material, about the content?
clarify, the gram iodine doesn't go in the fridge?
None of the stains go in the fridge. Crystal violet, iodine, safranin, and the small bottle of alcohol that's called decolorizer, do not put those in the fridge. The alcohol, it wouldn't hurt, but the other ones, don't put those in there, they'll precipitate. And then you'll have a hard time using them. All right, so nothing from chapters 8 or 9. All right, then, nothing from 1 through 5 that you're having on your quiz today.
I just have a question. So today, before I take it, I'm just going to study a little bit more. So you suggest that I just go back. I'm just going to watch the videos again and then, reread the chapters, right?
So I would suggest that ideally you would have read the chapters, watched the videos, taken notes, and then studied your notes, just like it was a regular test. The questions are not going to be in chronological order. They're going to be scrambled, and you're going to get a certain number of questions out of the whole large set. So there are more questions in there than you are going to get. So each student is not getting the same exact quiz. Mm-hmm. So there is 45 seconds a question. So there is not time to look up the answers. It's not supposed to be a homework assignment. It's supposed to be an exam. It's hard. I mean, I'm not going to sugarcoat it. It's not an easy test. I would say it's pretty challenging. Of the 5 people that have taken it, somebody already got like a 96. So, I mean, it's doable, obviously, if you've studied and gone through the material a number of times, but it is just like an in-person exam.
Okay.
So, there's no magic formula for how you should study for this. It varies from person to person. But like I said, the object of posting all those video lectures is for you to watch them and take notes. Because they are actually the lectures that I would have given if we were physically in person, right? So with a few things taken out. And I meant to put questions on there about the labs that you did, but I didn't get that done. So I don't know, maybe we'll have a lab quiz, maybe like one after the microscope work and then one at the end of the semester after the, actually after you've grown the bacteria and done those experiments with them, completed those experiments with them. So I have refreshed the Connect several times in the last couple of days. So it should be your assignments that have not shown that you've submitted them should be popping in and populating there in the coming hours, right? I'm going to say this again, even though I said it 100 times at the beginning of the semester, it is not instant. So if you are doing your homework assignments, like last night at 10 o'clock, and they're due at 11:59, first of all, they're not going to show up graded until, for some of you, until after the deadline has passed. And then they're also going to be a delay. So Connect their server has to talk to Canvas, and then Canvas has to actually dump them into my gradebook. And I'm not going to go in and look for you if your things have shown up, right? Unless there's a specific problem where they didn't show up after the due date, right? So Connect works a little differently than the Pearson set in that most of the time the scores aren't published in your Canvas until the deadline has passed or until the day of the deadline sometimes. Then I get a whole column that just populates with scores. So I had a couple emails this morning and I've put that in the announcement a couple of times. No, I'm not going to go double check if your grades, if you finished your work, right? That's kind of on you. You finish them until you get 100. And then if you finish them, you finish them. Right. I don't have time to go back and check 80 students and make sure that everyone who did their homework did it on time and send you an email. Oh, you forgot to do, or I say that, see that you haven't done chapter 4 yet. It's kind of, that's why they're all due the day before the test. So you have time to do that. And hopefully if you do them early enough, they will populate into the grade book But like I said, I don't have a lot of control over that. All I can do is go in every couple days and sync my section, and then it sometimes takes hours, sometimes takes a day and a half, and sometimes not until the assignment closes, and then all your grades pop up. So I'm reminding you of that again, because I've put it in the announcements and I've said it in a couple of the Zoom meetings, but apparently people still are sending me emails asking me if I can double check if their assignments are completed. And the short answer is not until after they're due. And then if yours don't come through, I will arrange for you to have an extension if there is a valid reason or excuse for you to need that extension, right? But pretty much they just come when they come. They pop in whenever that happens. Rather variable. Oh, I was going to check and see if people have already started to do chapter 8. Otherwise, I won't be able to. Oh, looks like it's still closed. Oh, good. So maybe I can fix that. Yeah, that's awesome. So I will shorten up the chapter 8. I will make it to be fewer, smaller bites. And I don't remember when I had these starting. It's probably after the quiz today. So maybe I can't. Let me just double check that one more time and see if I look at the assignment, if I can see when it's supposed to start. Let's see if it edits. Let me, kind of can't remember where it is in here. Yeah, that's unhappy. It's too many windows open at a time here. Yeah. Oh, there it is. Dang it. All right. Anyway, I will shorten those chapter 8 so that there's more modules to do, more homeworks, but they're not quite as long. Like I said, I'll try to see if I can manage the dates on that. Because I won't, let's try this guy. I hate when it doesn't show me what I've already selected. Okay, that's yeah, looks like it's not going to start until March 2nd. So I will cancel that. Just leave it like it was. I will moderate all of those assignments in that module so that they are open starting today, let's say. So I'm going to make a note of that. Connect, open all today. And then they will close just like these did the day before. So it'll be the 1st of March, right? On Monday, March 1st. For chapters 8, 9, and 6. Awesome. Okay, anything else before we have to sign off here? Yes, no? Anything else about the material?
I don't think so. On Tuesday, they'll have questions for 8 and 9 and 6.
Okay, awesome. All right, then I will see you guys on Tuesday.
Thank you. Have a great day.
You too.